I will present some of the genetic study on behalf of our CHOP group on the complex lymphatic anomaly, also include other vascular anomalies. So to let me go forward. Okay, here we go. So just a little bit background and history. So in 2015, we identified this germline variant in FNB4 in a patient with lymphangiomatosis. But the term was not used anymore today. So, and turns out the patient the proband has the central conducting lymphatic anomaly. So that's how I got into the field, and that's how I met Dr. Dellinger and Dr. Wakula discussing about the germline cause versus somatic cause. But anyway, we model this variant in our cellular model. In the zebrafish model, we confirmed this is a pathogenic variant, and then we published the paper. And very soon after, we identified a somatic cause for the central conducting lymphatic anomaly. For in 2 patients, we identify a exactly same ARAF variant in these 2 patients. And through the cellular model here with the primary and lymphatic endothelial cells and the zebrafish, we tried the because it's ARAF involved the MAP kinase pathway. We tried the trametinib, which is the inhibitor to the pathway. After we confirmed that inhibitor in our assays, we successfully applied the inhibitor to one of the patients, and as you can see here, the patient has significantly improved his clinical symptoms with the whole lymphatic remodeling and the reduction of the lymphedema. So then with all of this data and the existing infrastructure at CHOP, Dr. Adams and Dr. Harkness put together this comprehensive vascular anomaly program. A lot of things has happened, since then, and today I just be honored to present one of the projects came from this amazing program. So as we know, for now, there's a germline cause and somatic cause, as mentioned by Dr. Wakula. So we kind of aim for a comprehensive solution for this group of patients, and then we have we developed this tiered testing method with the exome sequencing, with the panel sequencing very deep. Then we can identify somatic variant with the even lower than 1%, for example, with the UMI panel. We sequenced to 80,000 sequencing depths, and then also because this group of patients, even though they have severe symptoms involved multiple organs, but usually the biopsy is not possible because of the risk bleeding risk and the persistence of effusion, kind of mortality and morbidity. So then we developed this method, try to comprehensively profile genomic profile this group of patients. So for a complex lymphatic anomaly, we do germline. We also do the somatic variant. And once again, since biopsy was not always possible, we try to get around this by using other materials that came from the standard clinical care. For example, the fluid drained from those patients. Otherwise, could be considered as a medical waste. So when we try to isolate the cells, we try to isolate cell-free DNA from the fluid, from the plasma, blood plasma. And then we apply this, our panel, try to identify the somatic variants down to very low liver fraction. So I've got there, so cell-free DNA refer to the DNA fragment that released in the biofluid by different mechanisms, for example, cell apoptosis, and by so we use this strict BCT tube to splice the cell-free DNA. Then we can take a look at it. Again, to get around the limitation of the tissue biopsy, we get the lymphatic fluid sample. We first centrifuge the sample, and then from the supinate we isolate cell-free DNA, and from the cell pellet we try to expand cells with the endothelial specific media to get some type of enrichment, and then we sequence those DNA. So for typical panel, I mean, some similar panel has been used in the cancer field. You just have a amplicons, PCR amplicon based panel. You have adapter. But in this case, even though you sequenced very, very high sequencing depth, you cannot get rid of the error that occurred in the, for example, NGS sequencing platform or the PCR error during the library preparation. So then we come up with a solution that we add the UMI sequence to the both end of the DNA fragment. UMI stands for the unique molecular identifier, which so here we use A base at both ends, and then we use this information. We can collapse the reads, then get rid of the random error that occurred during the process. And in this way, we can confidently call the variant from the collapsed reads family, as shown on the here. So we sequenced this panel really, really deep. As you can see, for majority of sample, we have a 60,000 sequencing depth. And then after the error correction with the collapsing the reads, we get effective mean read depth for majority of for majority of samples with the more than 1,000 effective sequence depths, then allow us to identify a variant allele fraction below 1%. So another challenging for the somatic variant research is also try to confirm the variant that you identify, because this is really, really low allele fraction and could be some artifact involved. So ddPCR has always been a good standard, for confirm the somatic variants, but it's pretty expensive because you have to design assay for every single candidate variant that you identified. So to get around this, we come up, we look at literature and we find this paper that published in 2017. They developed they report a blocker displacement amplification method, BDA. So basically, you design a blocker that perfectly match the wild type, and then you have a primer that compete with this blocker, and because of the mismatch in the mutant allele, you will have a different free energy. So here, in wild type is a positive, and in the mutant is negative, and in this way you can enrich of the amplification with the mutant allele actually, and it has been reported after 45 PCR cycle, it can enrich like 1,000 folds. So by using this way, we confirm all the somatic variant. But another beauty of this BDA assay that you can design one assay that can target a few different variant in the very close area. For example, here I showed the two blockers. This one can cover a few hotspot variants in PI3CA, and similarly in 1047 and 1043 residue. So here just a few examples to show, we confirmed every variant that we identified. In first case is about 1%, and we use this assay. We can show only mutant allele get amplified, and there's no wild type at all. And similarly, in another case, even lower, 0.15%, again only mutant allele get amplified. So the assay also can confirm those small indels. In this HRAS case, so we confirmed all the mutant expressed. And in a very unique case, we have two PI3CA variants affect the same PI3CA residue, 1047. And with this assay, we confirmed the two variants in one assay. As you can see here, it has 3 peaks. So in our cohort, we have 356 patients, and as you can see, it's a pediatric enriched, and sex are equally represented. It involved a few disease entities, but I want to highlight here we have 104 primary complex lymphatic anomaly. And here just a breakdown that we use, because we, as I mentioned, we use exome sequencing, we use panel, and with the exome we identified about 20% of the patient has a germline variant in primary complex lymphatic anomaly and other vascular anomaly. with deep. So we also did the deep exome sequencing at 500 folds, and we additionally identify variants for complex lymphatic anomaly. So with the panels, additionally, we identify 40% in the UMI particularly. So highlight the utility that we the panel that we use. Another breakdown here, just because we cannot always get tissue, as I mentioned. We try to increase the diagnostic yield by using FIP. For example, here we have 53% yield, and also from the cells that are enriched from the lymphatic fluid, we have additionally 38%. I mean, without if we don't do this, and those patients may lose the option for the medical therapy. And I want to highlight, cell-free DNA is a major part of our study. We confirmed 38% of those patients with the somatic variant. So for the primary complex lymphatic anomaly, as Dr. Wickham mentioned, rhabdomyopathy is a great contributor. So here we solved the 46% of those patients with 23 genes. But as you can see, they are kind of enriched in the rhabdomyopathy, both germline and somatic. But just a little bit over half of the individual has a somatic variant. 40% has autosomal dominant, and 5% have autosomal recessive. So for the complex lymphatic anomaly, there has been few genes has been reported in each category, as Dr. Wickham showed. But here, we with our assay and the panel and the cell-free DNA utility, we significantly expand the genetic landscape for the primary complex lymphatic anomaly. As you can see here, we identify some other known variants in the known genes. For example, in the GRA, we identify BRAF, HRAF, and the KRAS variant in GRA, et cetera, et cetera. So, and after that, we want to make sure those variants are pathogenic, and we did the cellular function validation as we did before, and just show you a few examples here. BRAF and the RAF1, we show the mutation. When we introduce the mutation to the primary endothelial cells, it increase the phosphorylation of ERK and also increase the lymphogenesis activity measured by here cumulated spotting and the number of spots. So with that, we to see if this could help patients to for the medical therapy, and indeed, in about 60-70 patients. It impacted their medical care. For example, in 43%, 25 patients, it support the current use of the inhibitor, and in 17 patients, it lead to change to another inhibitor because mTOR pathway or MAP kinase pathway, and another 38% either initiate or plan to initiate the therapy. And after a while, we monitor the outcome of the clinical improvement, and as you can see, majority of patients has improved the symptoms. 9 has a stable, and 3 has a mixed, and 4 are just too early to assess, and only 5% in 3 patients has a worsened symptoms. So with that, I want to summarize our this bench to backside study in vascular anomaly. We did the genetic profiling of the 356 patients, including 104 complex lymphatic anomaly. We expand the genetic landscape for primary lymphatic anomaly, and we provide this solution, to resolve the genetic etiology by using extremely deep sequencing and the less invasive sampling type, because we can use plasma cell-free DNA, and then we can identify somatic variant with the variant fraction down to 0.1%, and we have put together this knowledge base for future clinical trial along the emergency of the multiple inhibitors in all the pathways. So with that, I would like to thank all the mentors and the colleagues, Dr. Hackinson, Dr. Adams, Dr. Dory, and a huge thanks to Dr. Shepard, always working together and is discussing science. So and also the funding agency from R21, the Lymphatic Malformation Institution, and other internal grants. Thank you very much.